Hello and welcome to Nikolai's genetics lessons, and today's problem is the following three pieces of the sequences originate from the same DNA string and have been found by sequencing what is the sequence of the full DNA fragment. And early methods of DNA sequences included different techniques, and one of them were to break strand of the DNA. This can be chromosome. Chromosome represent one continuous molecule into smaller pieces. So let's say we break this string of the DNA into smaller pieces of the different size. It can be made by different methods, for example with the use of restriction enzymes, or we can do it using ultrasound. If we are going to use say the same restriction enzyme on the many similar chromosomes, actually chromosomes have to be identical, we are going to get the same size of the fragments, and we wouldn't be able to assemble them into continuous string. Actually, with sequencing technique, we also cannot sequence the whole chromosome because it is too big. But we can sequence smaller fragments. But if we are going to use the same restriction enzyme, we probably will get the same size fragments. And there are two variants whether we are going to use each time different enzyme. So we are going to get say next time, if we would use different enzyme on this chromosome, restriction enzyme, we can get in this case different size fragments, and in this case, as you see, we would have overlaps, and we can assemble such string of the DNA into one piece. But even better results we can get with ultrasound, because ultrasound would give us random fragments of the DNA. For example, if we would take, say, chromosome number 1 from the human karyotype, and if we would have 100 different variants of the same chromosome, and if we would break it using ultrasound, we are going to get every time different size fragments. That would have, of course, overlaps, and we can assemble them into one continuous string. Of course, we would be able to do it after we would sequence each small fragment, and machines usually would assemble them in such a fashion like this. So we should have overlaps, and then we would be able to tell the sequence of the whole fragment, starting from here and ending here. And usually when we use computers, we do not generate one such long continuous string, but we arrange into our sequenced DNA into contigs that also would have overlaps. And sometimes between these contigs we may have gaps. Then we would need more sequencing in order to connect these contigs. And here is going to be one contig, and here is going to be a second, and here is going to be a third one. So this is exactly what we are going to do today. We are going to assemble these three fragments into one. So we are not looking for fragments to be complementary to each other, but we are looking for fragments that would overlap and would have the same sequence. So here is the first fragment, second and third. Let's take a look if the sequences would overlap. And as you see, here we have overlapping, And this sequence, let's compare. We don't have such sequence here. Let's take a look here. And by the way, 5' end also have to be on the same side, and 3' end also have to be on the same side. So this is going to be another overlap here, and basically sequence of the fragment, full fragment, is going to be 5' end, A, G, C, G, T, T, A, G, C, C, G, G, T, A, and A, and 3' end here. So this is going to be our answer, and this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.